Hello children. So let us try to understand the last few topics of the chapter life processes. And today we are going to learn about blood pressure. The instrument which is used to measure it is sphygmomanometer and how it works, how it really measures the blood pressure. And then we will learn about lymph and a little bit about lymphatic system. So, what is blood pressure? The force that blood exerts against the wall of a vessel is called blood pressure. And you must remember that liquids and gases always exert certain pressure on the container in which they are contained. So if the blood is flowing inside the blood vessels, it will definitely exert some amount of pressure. And once the blood is leaving heart, then you can imagine that due to contraction of ventricle, there has to be a huge amount of pressure because this pressure will be utilized to send the blood throughout the body. Now, arteries are those vessels which actually carry the blood away from the heart. So, arteries are directly connected to ventricles and therefore they have much greater pressure in arteries than in veins. Also, the pressure during ventricular contraction, which is known as systolic pressure, is higher, and during ventricular relaxation or diastole, the pressure is a little bit lower. So let us have a look at the structure of heart and see which major artery we are talking about here. So remember, the left ventricle is about to pump oxygenated blood through aorta. Now this main artery is going to divide and subdivide into a number of arteries which will carry oxygenated blood throughout the body. But how am I ever going to understand what is the pressure over here? Because remember children, heart is protected inside the rib cage. So what do we do instead is just target any other artery down the network which is not inside the rib cage, and then we will be targeting that artery to understand what was the pressure that ventricular systole had given to the arterial blood. So, the standard location for blood pressure measurement is brachial artery. Let us see where does it reside and where do we find it. So, now this is the brachial artery, and if you look at the turn of your elbow, you can actually feel the pulsation at the inner side of the turn of your elbow. Just 1 inch above the turn, you will feel the pulse of the brachial artery. So how is sphygmomanometer works? What do we do children? If I am trying to obstruct or stop the flow of blood in this artery, whenever I will release the blood flow again, it will start flowing with the same pressure which the ventricular contraction had exerted before. So that's what we do. You must have seen that doctors always wrap around a cuff-like structure just above your elbow and they start inflating it. So once the cuff is inflated, it is pressing hard and very hard till your brachial artery is closed. Now you have obstructed the blood flow, children. So why did I do that? Because now that doctor is going to pay attention to the sound of the stethoscope. At this point, once the arterial flow, brachial arterial flow has been stopped, now the doctor puts his stethoscope to its ears and slowly releases the pressure in the cuff, slowly. And due to this slow release in the pressure of the cuff, brachial artery is about to open once again. But when is it going to really open? Exactly at that moment, children, when the pressure over here, this pressure which is continuously being monitored, equals the pressure with which your ventricle has pumped the blood, that is systolic pressure. Otherwise, If that pressure is any less, the blood will not start flowing. So we are slowly releasing the pressure, and exactly at the point when it equals the ventricular systolic pressure, the blood starts flowing. But then, since you had stopped the flow of entire blood through brachial artery, it is going to give you a sharp sound, tuck, and that sound heard at whichever pressure tells the doctor that okay, this was the systolic pressure of the patient. See, sound is audible in stethoscope because now you are suddenly allowing the blood to flow, and the blood will exactly flow with the same pressure with which ventricles had pumped it. And now the blood starts flowing and it gushes through the artery just like a river flows. But then, slowly, once the entire pressure is released and the brachial artery is almost in this situation, almost, then at the last sound is heard. A very mild and muffled sound is heard, and after which the sound stops in the stethoscope. That particular pressure point at which the sound stops tells us that this was the ventricular diastolic pressure. That was the pressure of ventricular relaxation. Okay, so once again, try to revise this concept. This artery is one of the main arteries running through our arm, known as brachial artery. Now, what we do? First, we inflate this cuff and stop the flow of blood, and we will start slowly releasing the pressure in the cuff. So there comes a point when the pressure in the cuff is equal to the pressure with which ventricles have pumped the blood, and that is when the blood will start flowing again. But that sudden opening of the artery creates a sharp sound which is heard by the doctor in the stethoscope, and the doctor measures that pressure at which point the stethoscope is giving sound. Then doctors hear some gushing sound, that is the usual sound of the flowing blood, and then a very mild and last sound. So that pressure when the sound stops is your ventricular diastolic pressure. The first sharp sound was ventricular systolic pressure. So what did we conclude here, children? The average pressure of ventricular contraction or diastole is about 120 millimeter of mercury. The higher mercury rises, higher the pressure is, and the standard or average ventricular relaxation or diastolic pressure is about 80 millimeters of mercury. And remember, stethoscope is an standard accessory of sphygmomanometer. It won't work alone. You will never know that at which pressure point you need to pay attention. Unless you hear this sound, you will not know that which pressure was the systolic or which pressure was the diastolic pressure. So I hope this is clear, and if not, you can ask me about your doubts. Now let us try to learn about lymph or tissue fluid. Lymph or tissue fluid is also known as filter fluid or interstitial fluid. So it is very interesting to know that how is this fluid created? What is the source of this fluid called lymph? For that, we must keep in mind the concept that we have just now learned, blood pressure. So when the blood is flowing through the arteries at high pressure, some amount of it leaches out through the interstitial spaces, and that space is not big enough to allow the exit of most of the cellular part, but the liquid component of blood flows out of the vessels and lymph is created. So let us see how is this going to be and where is this going to be. So you can see that this is one of the arteriole which is carrying the blood, and slowly some liquid portion of the blood comes out, and this is the tissue because capillary network will be surrounding the tissue or the cells. So if any liquid is coming out, that liquid is completely surrounding these tissue and cells, and that's how it is actually bathing them. So whatever it is carrying, that liquid, whether it is oxygen, whether it is nutrients, hormones, now they have direct access to all these cells. Even in those places where these capillaries are not reaching, the fluid part of the blood will reach in the form of lymph. So that is how first of all lymph is created. And then if this much fluid is coming out of the arteries while the blood is flowing, then it also needs to return back to the main bloodstream. Otherwise the overall volume of blood cannot be maintained. So there are various lymph capillaries which will take it back and drain it into the major blood vessels. So let us understand a little bit more about lymph. This is also known as filter fluid or tissue fluid because it is accumulating in the tissue site. It is not within the vessels, and it is formed due to filtration of the liquid portion from the blood vessels. Now, this point explains that how it comes out. So through the pores present in the walls of capillary, actually these are not pores but the space between adjacent cells of epithelium, some amount of plasma, protein and blood cells will escape into the intercellular space, and this will create lymph. Now, what is the composition? So it is obvious that if liquid portion or the matrix portion of the blood is coming out, the constitution of lymph will be pretty similar to the plasma of the blood, except that it contains less proteins. And what is mainly the cellular portion of the lymph is going to be lymphocytes. So its composition, other than somewhat less proteins as compared to plasma, will be very similar to it, and it will contain a lot more lymphocytes. And where is this present? It is present in the tissue portion. It is not present inside the vessels when it is doing its job. Once it has exchanged essential gases and nutrients with the cells, it will be drained back into the lymph capillaries. And what else is the function of lymph? Well, if you remember the lacteal present in the villi of the small intestine, lymph carries digested and absorbed fat. So that lacteal was exclusively involved in the absorption of fat from intestine. And also the excess fluid is drained back into the blood. So whatever excess fluid has been accumulated here, after proper exchange of gases and nutrients has taken place, it will go back and become a part of blood once again, and that's how the overall volume of blood remains constant. There are a few more important points about lymph. So, unlike the cardiovascular system, now what is cardiovascular system, children? Cardio means heart and vascular system means all that network through which the blood is pumped by the heart. So, unlike that system which consists of blood vessels and heart, lymphatic system is not a closed system. So remember children, our blood circulation system is a closed circulatory system. Blood is always flowing within the vessels. It is not floating around outside the vessels in normal conditions. And lymph is always floating outside those vessels. Also, the direction of cardiovascular system is cyclic. It exits the heart and it comes back to the heart. It exits the heart and once again comes back to the heart, and this cycle is repeated. But lymphatic flow is unidirectional. So in whichever place of the body the tissue fluid has accumulated, ultimately the lymph vessels are going to carry it only in one direction, that is towards the upper portion of the body, and that is where these vessels have to empty all that fluid into blood vessels. Similarly, the overall volume of the blood will be maintained. So lymphatic fluid comes out of the vessels while the blood is flowing inside them, but once its exchange job has been completed, it will come back only in one direction and will be added in the blood vessels. Also, its composition is very similar but not exactly similar to blood plasma. What else? Besides water, it contains some proteins, waste products that it has accumulated from those tissue and cells which it was bathing, and cellular debris and various pathogens. And why? Why not? Because the main cellular component of lymph is lymphocyte, and lymphocytes are very good at killing the pathogens, so that's why it also carries pathogens. And it contains lymphocytes as its main cellular component. That is pretty clear that why lymph is such an important fluid in our body. With this, I hope that the concept of blood pressure, how is it measured with the help of a sphygmomanometer, and what is the tissue or interstitial fluid, also known as lymph, is formed in our body, and what are its functions. And I hope that was helpful. If you have more doubts, please ask me. Thank you.